Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Biology. Today in this video lecture we are going to talk about vectors. Okay. In the previous video lecture, if you guys remember, we talked about the recombinant DNA technology, the big picture, the overview, the summary, the concept of recombinant DNA technology. Okay. We talked that what are the recombinant DNA technology. We talked the gene cloning, DNA cloning, as well as we talked that what actually we need in recombinant DNA technology. We also talked the steps of the recombinant DNA technology in detail. Okay, this is the second video lecture of the recombinant DNA technology, and in this video lecture we are going to talk about vector. But before explaining vector, you guys need to understand about the concept of recombinant DNA technology. Now if you missed that video lecture, it is present into my channel, you can watch it. Okay, or I will give the link in the description box, then you will check it. So let's start the vector. So let me write vectors. Now, what are the vectors? Vectors are the vehicles, or it is a delivery system due to which we can transfer or carry our gene of interest. So it is our vehicle to transfer target genes. Now whenever we want to discuss or explain the vector, that what are actually vectors, so for the identification of the vector we need to understand the basic properties of the vectors. Okay, so a genes should be called as vector when they have different properties, when they have basic properties like they have origin of replication, they have multiple cloning set, and they have selectable markers. Now these three are the main properties or basic properties of vector due to which we can identify that whether these genomes are a vector or not. Okay, now let me write usually there are different types of the vector: plasmid, cosmid, phasmid, cloning vector, expression vectors. But let me draw a simple diagram of the vector here. So here I will draw the plasmid vector, because plasmid is a very common vector which is used in molecular biology, or which is always used in the recombinant DNA technology. Although there are some other vector also used, but here I will draw the plasmid vector. So let's say if I draw the plasmid vector here, so how it will be look like. So all of you better know that vector are circular in type. Okay, and it is double stranded. Okay, as all of you better know that plasmid are a circular DNA. Let me write, it is a plasmid. Okay, so plasmid are the circular DNA having self-replicating capability. Okay, now for a vector, when we call the genes for a vector, they should have three main properties. The first property they have, let me write, they have origin of replications. Replication. Now what does it mean? Origin of replication means that a vector should have a specific region where a replication is started. So let's say at that specific area, a vector should have, let's say these are the origin of replication, origin of replication. Now vector should have a specific area where the replication is started. Now when the vector have a origin of replication, then we call that genes that it is a vector. When they don't have origin of replication, we don't call that it is a vector. Okay, so for a vector they should have origin of replication. Now one thing. The second thing is that with the help of vector we can transfer our gene of interest to the host cell. Remember, when we don't have a vector, so our gene of interest will be not inserted into a bacterial cell. When we insert the target genome or target genes to a bacterial cell without vector, so it will be degraded by restriction enzyme. Okay, but with the help of vector, our gene of interest will be not degraded, and it will be not degraded, and moreover it will be replicated, and we get multiple copies of our gene of interest. At the origin of replication, there are the start codon where the replication are started. Now the second property, a vector should have multiple cloning sites. So let me write. Short form we can write MCS. Now what are the multiple cloning site? Let's say at that specific area, a vector should have multiple cloning sites. MCS. Now multiple cloning site, or let's try to understand these words. First of all of you better know that cloning means to produce the same copy of the material that what we need. Site, the region, the specific area, and multiple means different. Okay, multiple means different. Okay, now what does multiple cloning site means? Now multiple cloning site, in case of recombinant DNA technology, multiple cloning site are those area where we can clone our gene of interest, but we can clone our gene of interest at multiple sites. At that area, at multiple sites of that area, we can clone our gene of interest. Why we call it multiple? Because at that specific area we can add a lot of we can add different kinds of gene of interest. We can add insulin gene, we can add some other genes, chaperone proteins, some other enzymes we can produce. Okay, that's why we call that multiple cloning sites. We call it multiple cloning site because we can treat this area with different enzymes. We can treat it with the eco R1, we can treat it with BAMH1, we can also treat the vector with the handy 3. Okay, so these are the three means there are not only these three, but there are different restriction enzymes which we used to cleave this vector at a multiple sites. Okay, now that's why we call it multiple cloning site. But remember, whenever we use as in the previous video lecture, if you guys remember, we talked for vector and as well as for the target genes. Whenever we want to join our target gene with a vector, we should treat our gene of interest as well as the vector with the same restriction enzyme. We don't use different enzyme. So when when gene of interest have multiple cloning site, we call that it is a vector. So that's the second property of the vector should have. Now the third property of vector should have, we call that selectable marker. Okay, now let me tell you that what actually happened once we insert our gene of interest into a vector, then we should transfer this vector to a bacterial cell, and once we transfer this to a bacterial cell, then the bacterial cell will divide it. But we directly not transfer it. We can transfer it when we treat the bacterial cell with a calcium chloride. Then we can transfer the vector to the host cell, and after some time the host cell will divide it, due to which our gene of interest is also divided, and finally we get our product. Okay, but it is not so simple as I discussed. Now after that, what actually happened, we have another property of the vector, we call that selectable. A vector should have a selectable marker. Now it doesn't mean that they have just like that marker. No, it doesn't mean that. Okay, now let me explain what does a selectable marker means. That at some specific area of the vector, there are some sequences where they can produce resistance genes. or antibiotic resistance genes. Okay, so they have antibiotic resistance genes. Let's say there are some sequences where the vector can produce antibiotic resistance genes. So these are called as selectable marker. Okay. Now why it is called selectable marker? Because let's say, whenever we want that which kind of vector, although there are many vectors, there are many gene of interest, there are many gene of interest, but some gene of interest are inserted into a vector and some vector didn't get the gene of interest. So how we can sure that which kind of vector can uptake our gene of interest? For that purpose we use the selectable marker. So selectable marker can they can express that your gene of interest is injected, or your gene of interest is added to the vector. Okay, now how we can identify that? We will what actually we should do for the identification of our gene of interest. We should make a petri plate and we should make ampicillin antibiotic petri plates. We should make those agar which contain ampicillin antibiotics, which are used as a selectable marker. We should grow each of the bacterial cell which have the vector, but those bacteria will grow when they have a vector, and those bacteria will die when they don't have a vector. Okay, now so when the bacterial cell are divided and grow, so we call that, so we make sure that this bacteria have our gene of interest. This bacteria will have the vector which contain our gene of interest. Okay, so how we identify that? How we make sure that? We make sure that with the help of this selectable marker. Nowadays we can use the fluorescences, we can use tag sequences due to which we can identify that the vector can carry our gene of interest. So due to selectable marker, we make sure that the vector can actually carry our gene of interest. Okay, that's the main point of the vector. Okay, in this case usually the selectable marker are the ampicillin resistance genes. Ampicillin resistant genes. Okay, now a vector should have these three main properties. Okay, now let me tell you some other basic important things about let me tell you some basic important things that what actually happens. Sometime what actually happen whenever we treat the vector with restriction enzyme, then the vector are cleaved. Okay, and when we add the gene of interest, as well as when we add the ligase enzyme, and when we add polymerase enzyme, and sometimes we add some other different kinds of material, okay, then what actually happen after that? After that, sometime this vector will self-annealing with each other, and there are three possibilities are usually happen during recombinant DNA technology, and due to which our experiment will be disrupted. Now what kind of three possibilities that are usually happen in recombinant DNA technology? Let's say we have a mixture. Let's say this is a test tube. Now inside the test tube we have a mixture of vectors. Okay, and with this mixture we have also our gene of interest. Moreover, they also contain ligase enzyme, and they also contain some other enzyme as well, restriction enzyme as well. Now what actually happen? We first of all we add the restriction enzyme, due to which they can cleave this vector at somewhere. Now the vector are cleaved by adding restriction enzyme, and when the vector are cleaved, then there are three possibility happen. The first possibility is that sometimes this vector are self-annealing. This vector are make bonding with each other when each of the ending are compatible to each other, so then they make a bond with each other. So the sequences are connected with each other, and we call that self-annealing. Sometimes this property happen. Sometimes each vector can uptake our gene of interest. So sometimes our gene of interest are also uptake, or sometimes what actually happen, the gene of interest which we inserted in the mixture, each gene of interest will make pairing with each other, or they make a pairing if there is a compatible and if there is a different or opposite sequences with each other. This is how these three possibilities are usually happen whenever we are doing molecular cloning. Okay, these are the main main problems which are happening. Sometimes what actually happens, sometimes when we treat the vector with eco R1 or BAM-H1 or handy, let's say any one restriction enzyme, if we treat the vector with any restriction enzyme, then sometimes they can cleave the selectable marker, or sometimes they can cleave the resistance genes, the ampicillin or the antibiotic resistance genes here. Once the antibiotic resistance genes are cleaved out away from that area, and let's say at that specific area we add our gene of interest, then what do you think? Our bacterial cell will grow or not? No, the bacterial cell will not grow. Why? Because the resistance genes is removed out by this restriction enzyme. Now once the means ampicillin resistant genes are released out, and once we transfer this vector to a bacterial cell, so then the bacterial cell is not divided when we grow it with the agar plate having ampicillin antibiotic, when they have an ampicillin antibiotic, so then such bacteria will not grow. Why? Because they don't have this resistance genes, they don't carry antibiotic resistance genes. So this is also another problems that are usually happen during recombinant DNA or during molecular cloning. Okay, so that's are the main problem that are usually happen in a molecular biology or in a molecular cloning. So that's are the main problem. Okay, so I hope you understand about the vectors. Okay, inshallah in the next video lecture we are going to talk about the gene library, and after that we are going to talk about the screening process. Okay, so that's about the vectors. I hope you understand. If still you have any kind of problems or confusion, you can write it into a comment. Okay, inshallah in the next video lecture we are going to talk about the types of the vector. Although there are two types of the vector, cloning vector and expression vector. We'll be talking inshallah in the next video lecture. So like this video, share this video and subscribe the channel, and also stay tuned because in the next video lecture we are going to talk about types of the vector. Thank you so much for watching.